Thank you, Ryan. First of all, I would like to thank organizers, SEC organizers, for inviting me to give this talk. And I would also thank a lot of my collaborators. As you can see here, it's a long list. I don't want to read. I will finish my 15 minutes with that. So these are the people from different bioenergy centers across USA and the various labs. And we have a bunch of people from BESC and a bunch of people from GLBRC, as you can see here. And the title of my talk is Comparative Glycomics of Plant Cell Walls or Plant Biomass, an insight into the composition, diversity, formation, and recalcitrance. It's a pretty big topic and cannot be finished in 15 minutes, but still, we will try to get some points to you through this. Now, as we all know, the plant biomass is one of the major sources for renewable energy, which we are talking about all these days. And the plant cell walls constitute the major part of plant biomass. And the problem with the plant cell walls, it's not a problem, it's a research problem with the plant cell wall is it's structurally very complex. As you can see here, there are two challenges in biomass-based research. One is understanding the cell wall structure dynamics in biomass from various plant sources. And the cell walls keep changing from plants to plant, biomass to biomass, and organ to organ, and even within different cells, it keeps changing. So it's very dynamics. So studying these complex structures, it is very difficult, and it's challenging to the researchers. And thus, we need to have a high throughput methodologies to rapidly analyze various biomass in order to get rapid know-how, how to utilize them downstream into the biofuel industry. Now, the second part is we have to elucidate the components affecting the cell wall recalcitrance. That means the innate ability of plant cell walls to resist deconstruction. You need to understand the components which cause that recalcitrance. Once we identify those components, it will be easy for you to utilize them to make bioethanol out of that. So that's one part. And the third part is we can analyze biomass which are modified due to pretreatment, enzymatically, chemically, and study their properties, how they behave in terms of their recalcitrance, and identify. So the first part, how to analyze such a large number of samples in a relatively efficient manner and in a high throughput or medium throughput manner to do that. So that's what I'm talking about. So what we need is, in this challenge, is tools. And one of the tools which we have developed in our laboratory is a comprehensive collection of monoclonal antibodies which are directed against plant cell wall glycans. As you can see in this figure, you can see we have a bunch of antibodies, around 200 antibodies, which is the largest collection in the world, which in this heat map, it shows their binding to a group of, or a wide variety of plant glycans. And as you can see, the largest, strongest binding is the yellow, and the dark color shows no binding. So these antibodies are grouped, and you can see there are antibodies against most of the hemicelluloses like xyloglucans and mannan and galactan, and we have antibodies against xylan, and we have antibodies against pectins, and we have antibodies against other things like beta glucans. So these antibodies, this collection of antibodies are really robust and comprehensive, and so that they can analyze most major glycans in the plant cell walls. So how do we use them? So we can use them to screen the biomass in a very efficient manner. So what we do is we take the biomass and make alcohol-insoluble residue out of it, which is basically cell walls, and fractionate the cell walls using a series of increasingly harsh reagents to get fractions which are selectively enriched in cell wall components, as you can see here. And we can load these onto a ELISA plate on an equal carbohydrate manner and screen them using ELISA, using the entire toolkit of antibodies. And we can do this in an automated manner. So there is an ELISA robot working in the lab, even when I am talking here. So this robot keep working and give you the throughput. So we can also do the experiments in 384 well format and get the data out of it and make the methodology more high throughput. So here is an example. We have analyzed some comparative glycomics of some dicot biomass. As you can see here, we have a group of herbaceous dicots and some woody dicots. And you can see here this on the right-hand panel, you can see the entire glycans detected by these antibodies. And you can always see that the top part is, represent the xyloglucan, then we have xylans, and we have galactomannan and glucans, and there is an acetylated mannan group, and we have pectins and arabinogalactans, things like that. So it's an entire spectrum of glycans in the plant cell walls, basically the non-cellulosic ones. So these components are really important. They are affecting the recalcitrance behaviors, and we can analyze that. So what we get? So we get some information like dicots analyzed so far lack xylan 3 epitopes. So xylan 3 epitopes, so based on the epitopes recognized by these antibodies, we can make conclusions about the structures of these hemicellulosic polysaccharides like xylan. So structural diversity of polysaccharides within the biomass we can analyze very efficiently using these antibodies. And similarly, the chloride extract. So during chloride extraction, what we do is we fragment the lignin, and whatever polysaccharides or glycans which are associated with lignin comes out or released in this extract, and we can analyze them using these antibodies. And you can see the chloride extract contain xylan, homogalactonan, ramagalactonan epitopes, but are devoid of xyloglucan. And from this information, we can say that xyloglucans do not have any interaction directly with lignin. And we know the glycans which are really interacting with lignin using this kind of analysis. And again, herbaceous dicot appears to have more arabinogalactan epitopes than do woody dicots. And we have analyzed some monocots. And you can see differing from the dicots, those xylan 3 epitopes, which define specific structures of xylan, are present in the monocot, which were not present in the dicot in the previous slide. So the biomass varies quite a bit, and these kind of techniques really can give us ideas like that. Again, the chloride extract, what is there in it, and there is another monocot species which we are working, which is a perennial monocot called agave, which is very promising in the field of bioenergy now, and the sugar yields are pretty high, and we analyze their glycome using this method. Mixed linked glucan, which is one of the prime component of grass cell walls, can be studied using this toolkit of antibodies. We analyze some gymnospermous woody plants like some Douglas fir and loblolly pine, and we analyze some dicots woody plants like poplar and black locust and things like that, and we get very nice information about their glycome. So what we need is we can also analyze comparative glycomics of grasses. So this is an analysis of 15 grasses. This is a huge amount of data. I cannot finish explaining this in a small talk like this, but I can just show you that if you take one part of this figure, like this chloride, post-chloride extraction, you can see there is a variation in the binding of these epitopes, showing that the glycome variation within the grass biomass. And are these variations connected directly to their recalcitrant properties? We need to study. Similarly, there are some antibodies binding differently to the different extracts, which we can analyze very in depth using this huge amount of data. So depending on the glycome, the recalcitrance varies. Here is an example. Here we have agave species which has a similar glycome as Arabidopsis, shows better sugar yield. And it is also similar to the Arabidopsis. The agave species are pretty similar to the Arabidopsis in terms of the pectic arabinogalactan epitopes, and they shows higher sugar yield. So lesser the primary cell walls, the more the sugar yield and lesser the recalcitrance. And that is not the case with the biomass with higher secondary cell wall components, as you can see in this case, the poplar and the switchgrass. So now we can analyze the pretreated biomass to get insight into the components affecting the biomass recalcitrance. For example, here is an experiment with poplar and switchgrass biomass. We have selectively done 3 treatments to remove pectin, to lignin, and xylan. And when we remove that, we get some extracts. some components are removed, we can analyze that using glycom profiling. And the residual biomass after removing these components, you can analyze for their sugar yield and get some data. So what we remove, we can analyze using the glycom profiling. For example, the chloride extraction remove these components in the case of switchgrass and these components in the case of poplar. So do the other extractions like base extractions and pectin removing enzymatic extractions like endopolygalactonase extraction and pectin methylesterase extraction. So you can analyze what is being extracted out and what happened to the residual, that we can analyze as you can see here. And what is in the residual, the residual biomass, you can always analyze their recalcitrance properties. So here is the sugar yield analysis of the residual biomass after all those analysis. So the nutshell here is, in switchgrass, you have to target xylan. If you remove xylan by base treatment, you can get a very high sugar yield in the case of switchgrass. As you can see, the KOH treatment shows pretty much 100% sugar yield in the case of switchgrass. But that's not the case in the poplar. In the poplar, you have to target lignin because the chloride treatment shows better response to the recalcitrance, reduced recalcitrance. So what component in what biomass need to be targeted, you have to do studies like this. Then only you can get a very nice productive downstream applications. So we can also analyze samples like hydrothermally pretreated. This is an experiment which we published in Energy and Environmental Science. And hydrothermally pretreated poplar plants show some difference. You can see the chloride extracts show reduced amount of epitopes present in them. There are multiple changes which we can pinpoint. When you pretreat, hydrothermally pretreat a biomass, what exactly happened to that plant cell wall components? To those, we can analyze those in detail using this kind of analysis. There are multiple informations we get, like easier extractability of xylan upon hydrothermal pretreatment and reduced pectic epitopes in the pretreated biomass. All those informations give us clues how pretreatments can be fine-tuned and optimized for better downstream applications. So do affects, which is one of the popular pretreatment method. In affects, affect the biomass in a different manner than hydrothermal. For example, I'll go back to the affects again. You can see that this is a bunch of data, very complicated, but in multiple, we have analyzed in the very diverse phylogenetic plants. What we need to know here is that if you look at the xylan, you can see that in the pretreated biomass, you have a higher abundance of xylan epitopes or higher binding of xylan-directed antibodies in the pretreated biomass in less harsh extractions, which is not there in the unpretreated. That shows that the non-cellulosic glycans are loosened out from the cellulosic core, exposing them to the microbial enzymes or other enzymes to act on them. So we have another type of pretreatment called alkaline hydrogen peroxide, which is also getting popular, and you can see an increased sugar release of alkaline hydrogen peroxide pretreated biomass. What is happening during that? We analyzed those biomass under alkaline hydrogen peroxide condition, and we found that in poplar and goldenrod, which is a herbaceous dicot, you can see the xylan epitopes are extracted out in the least harsh extractions. So do in the case of poplar. This is the same case in the case of grasses, where the xylan is an important recalcitrant component. You can see that the oxalate and carbonate extractions release xylan epitopes, showing that the reduced integrity of xylan in those biomass. So in nutshell, I made a model how to represent all this data. So we found that if this is the untreated biomass, in a hydrothermal condition, you can see that fragmentation of the non-cellulosic glycans, exposing the microfiber to the microbes and increasing the sugar yield. But in the case of affects and AHP kind of alkaline pretreatments, you can see that the non-cellulosic glycans are loosened out from the cellulosic core, making those biomass more in their sugar yield and reduced in their recalcitrance. So with that, I will go to the conclusions. And the glycom profiling based comparative glycomics is broadly applicable. It's broadly applicable in the case it can be applied in pretreatment. It can be applied in genetic modifications, or analyzing the genetically modified biomass. It can be applied in analyzing the pre-treated biomass. All stages of bioenergy research, you can apply this technique and get lot of information for companies and for research institutes. And I welcome collaborations, so please contact me if any of you are interested in doing that. And the second conclusion is the moderately throughput method assists in gaining insight into the diversity, composition, and overall structure of plant cell walls under varying spatial and temporal stages. And the third, while functioning as an excellent comparative glycomic platform, the study of plant cell wall biomass, the method can also efficiently monitor changes happening to them under varied circumstances, including genetic modification, chemical enzymatic treatments, and microbial conversions. So that's all from me. Thank you so much for your attention.
Questions? Again, get your hand up so the folks in the back can see you, and we'll get a mic to you right away. Again, I'll start with one. These techniques in glycomics, great for research tools. Do you think they would eventually be robust enough? You know, we choose our biomass on location, on yield per acre, on all sorts of different reasons why you choose maybe to locate a biomass. Ultimately, it's the cost per gallon if you're looking at biofuels. You think these would be really effective screening tools in maybe now or in the future to select a biomass?
Yes, I had several interaction with industry people, and they are like, they want to get some revenue flowing through the current biomass conversion industry and the biofuel industry, and their main aim is to do research like this to get better understanding of the components affecting recalcitrance. So I'm very sure that this technique is going to be used. Right now I am collaborating with at least 100 groups throughout the US and the world. So if I make a graph, it will go all over the world. So.
Okay, other questions? All right, thank you very much.
Thank you so much.